Thank you so much, everybody, for being here. I'm super excited about Friday Harbor/Lake Tahoe this year. So I'm giving sort of an overview talk, introducing some terms and concepts relevant to work we'll be doing on cognitive reserve this week. All right, so I have three major goals for this morning. The first, since we're at a methods conference, I thought I would try to organize and describe some of the most common methods that have been used to study cognitive reserve. And hopefully over the next few days, a lot of us will be elaborating on some of these basic frameworks. And then I'm very briefly going to introduce some terms that have been put forward to try to help explain the mechanisms underlying cognitive reserve. So here I'm really trying to distinguish between the sources of cognitive reserve, things like education, and the actual substance of cognitive reserve, the mechanisms, or what it is. And then finally, I'm going to share my view on how this concept of cognitive reserve relates to some of the other common terms that you'll see in the cognitive aging literature. So here, I hopefully will prompt some debate that we will almost certainly not resolve by Friday, but hopefully it will be worthwhile nonetheless. Okay, so in my view, a major impetus to study cognitive reserve came from autopsy data, like I'm showing here, which suggested that there are a lot of older people who exhibit high levels of brain pathology but never show clinical signs of dementia. So here you can see the brain on the right sure looks like the brain of somebody with Alzheimer's disease. I should mention that all these little black spots correspond to amyloid plaques. But in fact, this person never showed clinical symptoms of dementia in life. So in the 80s and 90s, several of these autopsy series were published, and they suggested that up to 2/3 of older people who meet normal cognitive criteria during life meet full pathological criteria for dementia at autopsy. So some of these larger numbers come from earlier studies that predate the standardization of quantitative neuropathological criteria for Alzheimer's disease, but even the later studies that use CRAD criteria still report pretty high numbers, so upwards of 45% of cognitively normal older people will show or meet full criteria for Alzheimer's disease on autopsy. So these observations led to a couple of questions, or maybe the same question asked in two different ways. So first, why is it that people with the same cognitive phenotype, so say people being non-demented, why do they differ so much in their degree of pathology? And yes, the other way of asking that question is that why do individuals with the same underlying pathology look so different on cognitive testing? And I'm going to argue that these two ways of asking the question have led to the two most common ways of looking at cognitive reserve in empirical studies. All right, so that first method of examining differing levels of pathology in the context of similar cognitive phenotype, there are some studies who have operationalized that similar cognitive phenotype as being in the same stage of dementia, so matching participants based on their cognitive level or stage of dementia. And other studies have operationalized this similar cognitive phenotype as a particular score on a cognitive test, so statistically controlling for a continuous cognitive score. So I'll give you some examples of each of these approaches to this method. So this is a really classic example of an early study that compared levels of pathology among individuals who all show the same or a similar cognitive phenotype. So here, Jakob Stern and colleagues recruited a group of individuals with mild dementia and examined levels of pathology across those individuals as a function of educational level. So here, pathology was indexed indirectly with this now old-fashioned xenon inhalation technique that measures cerebral blood flow. So here, warmer colors correspond to more blood flow, cooler colors correspond to less blood flow. So blue is sort of the proxy for pathology here. And you can see that people with more education, so in this group, the people who have more than a high school education, showed more pathology. So less cerebral blood flow in this temporoparietal region, more indirect evidence of Alzheimer's disease pathology. And the interpretation of these data was that these individuals with more education were able to maintain that same level of cognitive functioning despite their having more pathology. So this was one of the early ways of demonstrating that education is associated with cognitive reserve. Now here's another example of that same approach of comparing levels of pathology in the context of similar cognitive performance. But this study differs in that instead of matching individuals based on cognitive level, this is from the YCAP study, recruited a diverse group of individuals with differing levels of cognition and then statistically controlled for cognitive level in this structural equation model. But the finding was the same. So specifically, at any given level of cognitive performance, having more education or a higher reading level was associated with more pathology. And pathology in this case is white matter hyperintensities. So the interpretation is the same, that these individuals who have more education and higher reading level were able to perform well on those cognitive tests despite their having more pathology than people with less education. So the other way of asking this question, instead of asking why do people with the same cognitive phenotype differ so much in their underlying pathology, we can also ask why do people with the same degree of pathology differ in their cognitive status. And the primary way individuals have answered this question is by testing for moderation of the pathology-cognition association. And indeed, some would say that this is the strongest test of the reserve hypothesis, that some proxy variable moderates the pathology-cognition association. I'm also putting in this category the residual method for quantifying cognitive reserve. We're going to hear a lot more about this method later today and tomorrow. There's a tutorial about it in the evening. But here I'll just preview and say that this method involves isolating cognitive variance after partialing out or controlling for pathology. So here is a classic example of a study that looked at how education moderates this association between pathology on the x-axis and cognition on the y-axis. This is using data from Rush University. You can see that people with lower education, so this is the dashed line, these people showed a stronger negative association between pathology and cognition. In contrast, people with higher education, so this solid black line, high levels of pathology were not as strongly associated with lower cognition in this group. So this is a similar study that used this same method, only incorporating longitudinal data now. So what I've shown you so far was purely cross-sectional. Now we have our first longitudinal study. This is a super recent paper from Dan's group. And here in this first panel, you can see we have now time on the x-axis, and time corresponds to the accumulation of pathology, or in this case, gray matter atrophy. And cognition, again, is on the y-axis. So again, we see that people with less education in blue show a stronger negative association between advancing pathology or brain atrophy and cognitive decline, whereas you don't see that association in people with higher levels of education. Now, what's interesting in this paper is that when you looked across groups who were defined on the basis of their average rate of atrophy or their dementia stage, this relationship actually lessened and then eventually reversed, such that if you're looking just at people who have a lot of gray matter atrophy, so people who are characteristic of having dementia, education is associated with the opposite relationship. So now it's people with higher education in red who are showing this stronger negative association between gray matter atrophy and cognitive decline. And this pattern of results is actually in line with cognitive reserve theory. So this is shown in this is theoretical data here. You can see that having more education or having more cognitive reserve is protective early on in the disease process, so early on this continuum of neuropathology, in that it allows individuals to maintain their level of cognitive function for a longer period of time, so delaying this change point that signals the onset of cognitive decline. But at a certain level of pathology, so as you go along this x-axis here, it sort of catches up to you. And people with more cognitive reserve can show an accelerated cognitive decline. And this sort of theoretical model has been borne out in many, many empirical studies showing that individuals, once they develop Alzheimer's disease, those with higher education decline at a faster rate. They show a faster clinical progression. And this is really demonstrates one of the challenges of studying cognitive reserve, that it appears that it can be depleted, that it's a dynamic construct. And so studying cognitive reserve just by using these static proxy variables that don't change, so things like education, can be kind of unsatisfying. And that leads me to this residual approach. I had to show this crazy model because it came out of the Friday Harbor Conference, I think, in 2010. So this is a very typical model that we'll all be running in the next several days. So just to orient you from this model here we're using data from Rush University again. This model essentially takes a whole battery of neuropsychological tests and regresses them onto all of these neuropathology measures, so all of these quantitative variables obtained from autopsy data. And then the residual, so cognitive variance that's not explained by pathology, is separated out. And this study was interested in looking at what types of variables, so things like education and cognitive activity, socioeconomic status, what variables are associated with that cognitive residual after controlling for pathology. So this is basically the flip side of that structural equation model I showed you a few slides ago from the YCAP study that was looking at the association between education and reading and pathology controlling for cognition. Here we're looking at the relationship between cognition and education controlling for pathology. So it's really the flip side. Yeah, so in this study, variables like having more education and reporting more cognitive activities at age 40 were associated with more residual cognitive variance. So more cognition even after controlling for level of pathology. All right, so I'm going to move on just briefly to introduce some of the potential mechanisms of cognitive reserve. So what is cognitive reserve? We often talk about cognitive reserve in terms of education and activities, but those are really things that we think build cognitive reserve or contribute to cognitive reserve. Those things aren't cognitive reserve itself. Cognitive reserve really refers to aspects of how the brain processes information, so aspects of the brain's software rather than its hardware. So just in a few slides, I'm going to introduce a couple of these terms, but I think in Dr. Stern's talk tomorrow, we're going to hear a lot more about these terms. All right, so I have to show it with a picture. So here are a couple of terms that have been used to describe characteristics of brain networks, efficiency and capacity. And here I'm showing you an image of differing levels of efficiency in a brain network in the context of the same level of capacity. So here, brains that are less efficient or networks that are less efficient tend to ramp up activity faster early on in this continuum of task difficulty, and then they reach their individual capacity limit sooner than brains that are more efficient who take longer to reach that capacity limit. So in terms of capacity, brains that have higher capacity are able to generate more network activity than brains with lower capacity. So both efficiency and capacity refer to characteristics of individual brain networks. Another potential mechanism of cognitive reserve is this concept of compensation, which instead of talking about aspects of an individual network, compensation refers to sort of the coordination of activity across networks. So here in red, I'm showing you activity in a primary network, and by primary network I mean this is the brain network that's most typically brought online in service of a particular cognitive task. So as a task becomes more difficult, activity in that primary network ramps up and eventually reaches its individual capacity limit and sort of plateaus. But if this brain is flexible, it may be able to bring online other networks that aren't typically used in service of that cognitive task, but at certain levels of task demands could be brought online to support that primary network or maybe even to take over for that primary network at very high levels of task demands. And so compensation is inherently related to these concepts of efficiency and capacity because how efficient and how much capacity this primary network has dictates if and when these alternate networks need to be brought online. So I'm just going to summarize all of that in words. I much prefer pictures, but I know some people prefer words. So these first two terms, efficiency and capacity, again refer to aspects of a primary brain network. You might also see the term neural reserve used, which is used to describe these types of characteristics of individual networks to differentiate them from the concept of neural compensation, which refers to sort of interactions across multiple networks. Okay, so enough task-based fMRI. We're not going to be working with task-based fMRI data in the coming days. And the last part of my talk, I just wanted to introduce some very similar terms that are used in the cognitive aging literature and kind of share my view of how the concept of cognitive reserve compares and contrasts with those terms. This is a big point of debate. So the first term I wanted to talk about was brain reserve. So this obviously sounds very similar to cognitive reserve. And insofar as both brain reserve and cognitive reserve are interested in explaining why there could be an absence of cognitive impairment in the presence of pathology, then they are very similar. But whereas cognitive reserve is typically used to talk about characteristics of brain networks, so how efficient and how flexible brain networks are, so characteristics of the software, brain reserve is more commonly used to talk about characteristics of the brain's hardware, so quantitative structural aspects of the brain, like how big the brain is, how many neurons there are, because those structural aspects of the brain dictate how much of an insult or how much pathology an individual brain is able to withstand or tolerate before showing symptoms. So an analogy here is if you have $10,000 in your bank account and you get a $100 speeding ticket, your quality of life is probably not going to take much of a hit. But if you have $50 in your bank account, then that speeding ticket is probably really going to change your lifestyle. So Paul Satz, who was a pioneering neuropsychologist, first introduced this term, I believe. And he described brain reserve as a threshold model. So people have different thresholds for how many neurons they can lose and still have enough tissue left to support cognition. So I'll show you a picture from his classic paper. The terminology that's being used here is stroke terminology, but I think the same ideas apply to the dementia world, that any given level of pathology, so any given level of loss of neurons, can lead to different functional consequences for individuals that had different thresholds or different amounts of brain reserve. So for some people, that same level of pathology might not be enough to make them drop below some arbitrary functional impairment cutoff. So in our case, this functional impairment cutoff can be thought of as dementia. Another concept I wanted to talk about is brain maintenance. And this is a term, I think for clarity, I'm going to propose that this term overlaps with terms like neuroprotection and resistance. So brain maintenance talks about being resistant to the accumulation of pathology. And I think one reason why brain reserve and brain maintenance can get confused is that a lot of the studies that we do are cross-sectional studies that measure pathology indirectly through something like brain volume. And if you take a snapshot of somebody's hippocampal volume at one point in time and find that this person has hippocampal volume in the 10th percentile, we don't really know if that high level of hippocampal volume reflects the lack of atrophy, so brain maintenance, a lack of accumulated pathology, or if that individual just enjoyed, an exceptional hippocampus for her whole life. That would be brain reserve. So I think that's one reason why brain maintenance and brain reserve can be confused, particularly in cross-sectional volumetric MRI data. So this is an image from that original Nyberg article that put forth this term of brain maintenance. And the point of this figure is just that there are vast individual differences in older adults' susceptibility to amyloid deposition. So here, if you look at the older group, a lot of older adults don't have any more amyloid in their brain than middle-aged adults. But other older adults seem to be much more susceptible, so these red dots. So these black dots here on the right, these are older people who don't show a lot of amyloid. I think that this could also be a pretty heterogeneous group. So this could include people who don't have amyloid pathology because they have no risk factors. They have no genetic risk factors. They don't have ApoE4. But there might also be people in here who have a lot of risk factors, who have 2 copies of the E4 allele, and yet still don't show amyloid pathology. So I think that those 2 groups of people are interesting to differentiate, and that brings me to this concept of resilience. So resilience is a term that has a long history in the psychological literature in describing the process of adapting well in the face of trauma or adversity. So in the psychological literature, somebody might be in good mental health because they never experienced any trauma or adversity, or they might be in good mental health because they were able to bounce back, or they were resilient to some trauma or adversity. And so I think the same can be said for both brain pathology and cognition. So somebody can have a very healthy brain because they have no genetic risk factors for pathology, and somebody could have very good cognition in part because they have no pathology. But can we call those individuals resilient? I think inherent in the psychological use of this term is that it can be difficult to really observe and measure the process of resilience before adversity or before the accumulation of risk. We can measure perhaps people's potential to be resilient in the form of proxies like education, try to predict how resilient they would be if they had risks or pathology, but it's difficult to really observe that process of resilience. And resilience really is a process of adapting. So notice that I've used sort of 2 examples of the adversity analogy in cognitive aging research. So one type of adversity is risk factors, so genetic risk factors, and another type of adversity is pathology itself. So here I think that it might be helpful to distinguish between brain resilience and cognitive resilience. So these are terms I know that Dr. Homan has used these terms in papers before. So Tim Homan thinks a lot about phenotypes, and I think that phenotypes can be defined at different levels, at the brain level and the cognitive level. So brain resilience might be having a healthy brain despite genetic risk factors or other health risk factors, whereas cognitive resilience might be having better cognition given some level of brain pathology. So I think that these are worthwhile terms to use and to differentiate between both in our empirical and our theoretical models, because I think they have implications for understanding the mechanisms of health and disease. All right, so I just want to sum up some of the major points. So first, since this term or this concept of cognitive reserve was really born out of the mismatch between pathology and cognition, I think we really need data on both pathology and cognition. So it's difficult to study cognitive reserve if you don't have both types of data. And at the conference this week, we have access to 3 exceptional data sets that have both brain data and cognitive data. So I think this is a great opportunity to start modeling cognitive reserve. Another point is that cognitive reserve really refers to the active coping with pathology. So it can be difficult to observe it or measure it in the absence of risk factors, in the absence of pathology. I think there might be models to try to push people to show reserve by parametrically varying the task difficulty or something like that. But for most studies, it's really difficult to measure unless you start seeing pathology. And then the last point I want to emphasize is this idea that cognitive reserve is a dynamic construct. And this aspect of cognitive reserve can yield different patterns of association if you're looking at different stages of disease, and really seems to be a challenge for our modeling. I think that indeed probably the supreme challenge of studying cognitive reserve is that all of the variables we're interested in are all changing simultaneously and in relation to one another. So age is changing, pathology is changing, cognition is changing in response to those other variables. So this conference is a really great opportunity to start measuring changes in all of those constituent variables and multivariate and longitudinal frameworks. All right, so that's all I have. So thank you very much, and I'm happy to take questions.
We have time for a couple of questions. Yeah, thank you.
One question I have is when you say that a certain pathology indicates a certain disease, then I guess you would, if we started seeing results like, oh, well, all of these people have this pathology and they're not exhibiting cognitive signs of the disease, then maybe when do you start to question that your definition of the disease is incorrect?
Yeah, yeah, I think that some diseases, it can be easier to talk about reserve. So in the case of stroke, I think we all agree that having a lesion in the brain is what's causing impairment, and we can look at individual differences in how people are performing despite, the same size of lesion in the brain. But I think this is another reason why the disease that we all study is more difficult. So to what extent is this resilience or a misclassification of pathology? And I think what adds to that problem is that usually how we measure pathology isn't direct. We're using hippocampal volume or cerebral blood flow or resting fMRI or things that we're not totally sure how those variables relate to the pathology. We're getting better at measuring the pathology, but again, I think it may not be what's driving impairment.
Just from that, I think in the beginning when you were first talking about, they saw there was these brains that were filled with amyloid plaque, yet the people were perfectly cognitively normal throughout their lifetime. And it's interesting that then, I'm sure work has gone into that, into different paths of that too, but it's interesting that now we're trying to explain that instead of saying, well, wait a second, maybe that amyloid plaque isn't what we should be looking for.
Yeah, it's a really interesting history with the autopsy data because, at first those percentages were really high. So many people are normal and they have this pathology, and so then they started refining the standardization of that, and how do they refine their criteria? They do it by having the gold standard be impairment. And so naturally, the proportion of people who have pathology and don't have impairment goes down because they start defining pathology based on cognitive impairment, which makes a lot of sense. But even taking that approach, we've not yet succeeded entirely in perfecting those criteria. So I think part of this is we need better perfection of our pathological criteria. Yeah. Okay. Excellent question. Thank you, Teresa.
No doubt all of us in the room are delighted that higher education is protective. To a point.
So it's a compression of morbidity.
My question is, education is really a very amorphous term.
Yeah.
So what is the essence, or what is the impact of education, and what does it really mean to have higher education? What kind of cognitive activity is going on to make it protective?
Yeah, that's a great question. I think this is one of the questions that's going to be motivating Work Group 1. So shout out to Work Group 1. Most of these studies have really just looked at education in terms of years of education. And that means something very different across people, depending on what that education looked like and what they did with it after. I think when we're talking about the mechanisms of the protective effect of education, they're very broad. I'll show a study in my next talk that tries to look at socioeconomic status as a downstream effect of education. That might be one mechanism that really doesn't have much to do with brain function at all. It's more to do with sort of economic resources and access and that sort of thing. But education might also instill certain cognitive strategies that do shape the brain. I think educational neuroscience is a really growing field that has been looking at how different educational experiences rewire the brain. But it's a very young field, so we don't totally know. The other thing I'll mention about the study I'll show shortly is that education is also so different when we're talking about a year of elementary school versus a year of college. Not only are the activities you're engaged in during those two different types of educational experiences different, but you're at a different developmental period. So we don't know if differences between early and later education reflect sort of what's being done in the classroom versus what the critical period is that the brain is in when you're that age. So these are excellent questions that I don't have the answer